A DNA molecule has a certain sequence, and there are a variety of proteins or RNA molecules that will bind to specific sequences in DNA. So we talked about promoters before. A promoter has a certain sequence, and there's a certain specific set of proteins that are able to recognize that sequence and bind to it. So each, I guess, variety or class of promoters has its own sequence, and then there's a variety of transcription factors that bind to each sequence. This is just general transcription factor, which is a class of transcription factors. So you can see there's a few types down here, and they form a complex at the transcription initiation site, or upstream of that. So here's another transcription factor. Just another transcription factor in that complex. So this one here is binding to a Tata box, so it's looking for a stretch of repeating thymine, adenine, alternating bases. Those are kind of the weaker of the bonds. The CG bonds are stronger than the AT bonds. So the complementary strand is also A's and T's, and so you get a kind of a weaker force holding those two strands of DNA together, which makes it kind of easier for the protein to kind of, I guess, one, to pull those strands apart, but two, also maybe to be able to kind of make its way into the middle of the DNA to kind of probe which bases are there, maybe even. So here's still more of just the complex that forms. This is all specific to transcription initiation. But there's 100 proteins are found, in the ballpark of 100 proteins are required to form this complex. So it might not just be a single protein that's recognizing a Tata box or any other sequence. There might be kind of a combination of proteins that actually fit together and recognize the total sequence. The homeodomain proteins also are transcription factors, and kind of bond to some similar, instead of Tata, it's a TAAT kind of a sequence. But still there's a protein and it's able to match to a certain nucleotide sequence in the DNA. The zinc fingers have a zinc ion attached to a protein, and there's a variety of transcription factors that probably use the positive charge on the zinc ion to attach to the negative charge of the phosphates on the DNA backbone. There's another set of proteins that use RNA to determine, well, they use RNA as a way to match a specific DNA sequence. So here's the Cas9 protein from the CRISPR-Cas9 kind of gene editing complex, or whatever you want to call it. So this Cas9 is a protein which does its job of cutting the DNA or nicking it, and it uses the guide RNA as a means to match the DNA. So here you can see the protein with a guide RNA which is what gives it the specificity of matching a DNA sequence. And I guess in this case, you have a protein that does a certain job, and you kind of reconfigure it with different RNA molecules to match different sections of DNA. A similar way, the telomerase enzyme, I think it's an enzyme, actually it's reverse transcriptase, I think, yeah, reversed transcriptase enzyme. So it's using another kind of like a similar guide RNA to then build more DNA on a telomere. And then you have something like a transfer RNA, which is an RNA molecule that has its own folded secondary and tertiary structure, and is acting itself as kind of an enzyme. It's not relying on a protein to do the work. The RNA molecule does the work itself, and it's using a few of its sequences to match to a messenger RNA, and then using its shape to work with a ribosome, and also bind an amino acid to it. So that's kind of an example of an RNA molecule doing what normally would be a protein. As far as finding a specific RNA sequence, and also doing the job of, in this case, bringing along an amino acid to add to a chain of growing amino acids on a polypeptide. And just a kind of another side note is this chromatin immunoprecipitation is a way to kind of freeze the proteins to the DNA, or lock them in place, and then you're able to chop up the DNA, use the immune system, but it's the antibodies. So you have specific antibodies to bind to specific proteins, and you can add probably like a bead or something to give them a little bit more mass. And then once you have a protein with a antibody attached to it with some mass, you can use a centrifuge to filter out the heavy material, and then remove the proteins and sequence the DNA that was captured. This way, like with a transcription factor, you could filter out all the DNA that was underneath a certain transcription factor to kind of see what sequences you're finding in a transcription factor, or sorry, to find out the sequences for a promoter or enhancer or anything else that is getting a protein bound to it. I think it sequences what's underneath the protein, but there's a chance it sequences what's on the sides of it, and then you have kind of a gap here, then you can compare that to just the straight up DNA sequence to find what's actually underneath the protein there.